But let me start with just the bigger picture of aging. So aging is, clearly something that we easily recognize, but actually find hard to define in terms of molecular processes, physiologic processes. I mean, basically what we know, what we would all say is there's some element of decline in structure and function that is the prototypic characteristic of aging. So even though we can't define it so well, we certainly are interested to find out if there's anything that we can do about it. So I'll talk a little bit about kind of interventions that are very high level that have become of interest in the fields of aging and GR science and are clearly of interest to the general public in terms of what we can do to potentially slow, arrest, and maybe even reverse aspects of aging. And this ranges from things like drugs to diet and obviously to exercise. So let me just begin with a couple of famous drugs that you've probably heard of that have been studied in this context. And one is, of course, the drug Metformin. And many of you will have heard of this. It's a very common diabetes drug. And it's from a class of compounds that were originally found in the French lilac and used really in folk medicine for several centuries. And it's quite effective in treating diabetes. In studies of the pathways that are affected by this drug, there became an interest in how this kind of generally shifts metabolism. And investigators began to look at this in terms of how it affected aging in the kinds of animals that we study in the aging field, which include worms and flies and mice and so forth. And it was found that treating these animals with metformin could actually extend their lifespan. And that became kind of almost this kind of prototype of asking, if it extends lifespan, might it actually extend health span in humans, is where the field has evolved. But a lot of this has really evolved around looking at lifespan extension. But there was a key paper that was published now many years ago, quite a few years ago, from the UK, looking at the effects of metformin on survival in humans. And these are what you see below here is what called a kind of survival curve or Kaplan-Meier curve, where you start off with a group of people, all of them are alive, and then you follow them and some of them begin to die. And that's how this curve falls off here. Now, these are something 70-year-old people. And so you see over this time course of several years, people begin to die. And these curves here, the black curve and the red curve, are healthy people as they begin to die. In the blue line here are patients with diabetes being treated with a different drug. And as you can see, their risk of dying is higher. More people die over a shorter period of time. But what was really interesting and really surprising is that this green line here are patients with diabetes, but treated with metformin. So this is striking because not only does this drug reduce the risk of patients with diabetes from dying, but it actually looks as if, small study, that they actually survive better than patients without diabetes. Okay, so this really, from a human epidemiologic standpoint, really raised the question of whether this kind of drug might be actually slowing the aging process in addition to treating diabetes in a way that might be good for everybody in a general sense. So that was the concept, and that idea is out there, that there may be drugs that can actually slow the aging process and that might be beneficial to the population at large, or at least a group of people that could actually be given to people before they develop diseases and that could actually extend their lifespan by preventing the onset of age-related diseases. And this drug, metformin, is actually being considered by the FDA now for a clinical trial that will be, in a sense, the first of its kind. That is a drug that actually is being used to treat aging itself, not a specific disease, but treating people and looking for the ability of this drug to delay the onset of these age-related diseases. Okay, so this is a paradigm shift, and it's a big issue for the FDA, appropriately so, as to whether we should be developing drugs that are not for specific diseases, but are actually for this process that we're very interested in studying, this process of aging itself. So this trial is underway and designed, and we'll find out more about it. So the next drug I want to mention is Rapamycin. And of course, this is a drug that Joan knows very well. She studied it for many years. And likewise, this is a drug that was found for another reason. It was found on Easter Island, a Chilean island, so-called Rapa Nui, and that's where the name Rapamycin came from. And it was found initially to be basically an antibiotic, I think an antifungal actually. And but then it was found to have very strong immunosuppressive properties as well. And now it's used commonly, for example, in organ transplants. and other treatments that involve some kind of adjunct immunosuppression. Well, in the same way, this drug started to be understood in terms of the proteins that it interacts with in the cell, and it became very of interest as to how this drug, rapamycin, actually interacts with cellular metabolism as well. And in the same way, investigators started to study this in model organisms. And I think the first evidence of this drug as being kind of a lifespan extension actually was in yeast, but then in other model organisms in flies and worms, and had quite a potent activity in terms of extending lifespan. So that's, again, another important kind of avenue, looking at drugs that might be used in this way as modulators and perhaps slowers of aging. But probably the granddaddy intervention for slowing aging is caloric restriction, so withholding calories. And this has been around and known since the 1930s. Here's a graph of kind of a lifespan curve. This is in mice. And so here again, all the mice are alive. In the green, you see the control mice. They begin to die. About half of them are still alive around 30 months of age. Well, if you restrict the calories by 25% or massively 55% or 65%, you see the animals live longer and longer and longer, and almost a doubling of lifespan by this really massive caloric restriction in these mice. So this has been replicated in lots of species, many, many species. There's strong evidence that this occurs in primates even, that you can actually delay the onset of death, delay the onset of age-related diseases by restricting calories. And if you're so inclined, you can join the Caloric Restriction Society, and you can restrict your calories, and you can be hungry for a very long time. And if you don't get hit by a bus, maybe you'll live longer. We don't know in humans yet. There's certainly evidence from studies of fasting and other dietary interventions that there is likely to be an effect in humans, but we don't have any actual lifespan studies. So we came into this field of interventions for affecting aging in a kind of roundabout way, and we came into this really from our studies of stem cells and tissue repair. And our interest came, we were originally just interested in how tissues repair and the stem cells in the body that repair them, but we became interested in the question of why do we lose a lot of this reparative capacity as we get older. So in a younger human, if you injure a tissue, a skin or a bone, they heal very, very well, very, very quickly, and with very little scar formation. But as we get older, we all know this, that our wounds, our injuries heal more slowly, often with a lot of scarring. And we know that this is not due per se because we run out of stem cells. We still have plenty of stem cells, but the stem cells, and this is something we've studied in detail, begin to lose their efficacy in terms of tissue repair and tissue regeneration. So the tissue that we've studied primarily is skeletal muscle. And I'll just show some basically representation of how good muscle is at repairing itself. So just like skin. So on the top here, these are a microscopic view of a muscle from a mouse. And then after a few days of a total necrotic injury to that tissue from a toxin, you see a destruction of that architecture. And after a few days, you start to see the formation of these newly forming kind of muscle fibers that over the course of a few weeks grow and restore that tissue. So really this remarkable regenerative capacity to basically go from a totally damaged tissue back to a normal tissue. Well, that capacity does decline with age, and this is illustrated in this slide from some data from our lab in the mouse. And this has been known before we did this, but we're just illustrating it here. So these are muscles from a young mouse or an old mouse. Here's the uninjured muscle. After a first injury, a second injury, you still get very, very good repair. But in the old animals, even after a single injury, you start to see these areas, these sort of whitish areas that are the beginning of scar tissue formation. After a second injury, you see a lot more scar tissue formation, and some of this blue area here is more inflammation. So just the ability to repair is something that we see declines with age. So we became very interested in asking, can we intervene in a way to restore this youthful reparative capacity to old tissues and old animals? And we had evidence to support the idea that some of the enhanced repair in the young animal comes from positive reparative factors in the blood, and some of the declining regenerative capacity with age came from suppressive activities in the blood. And this led us to do this very strange, but turning out to be a very important experiment are using this technique called parabiosis. And parabiosis is a technique whereby animals are surgically connected and they develop a single shared circulatory system. So the tissues of one animal are exposed to the circulating environment of the other animal. And these kind of control pairs are so-called isochronic, so same age, young animal to young animal, or old animal to old animal. But the interesting experiment of the so-called heterochronic parabiotic pairs, where a young animal is connected to an old animal. So these animals, again, develop this shared circulatory system. And the old animal, over a few weeks, begins to be exposed over and over to the environment, to the circulation of this young animal. And we ask the question, what effect does that exposure to this young blood have on this old animal, by then looking at, for example, its muscle stem cells and its ability to respond to a muscle injury? So we did these experiments where we would introduce an injury into either the control pairs or these heterochronic pairs. And what we found was really quite striking. So in these control pairs, this is an early phase of regeneration. This is, take my word for it, very healthy regeneration. In the old partners, young, old to old, we start to see a lot of this fibrosis that I mentioned, a lot of scarring. But in the old animal that had been connected to the young animal for some period of weeks, they repair just as well as the young animals. So, and we were able to find, we've done many more studies on this, that there's really a restoration of the stem cells that engage in this repair from an old state to a young state. So that we really seem to be reprogramming, in a sense, these old cells to become younger. So is this truly rejuvenation? And that's the kind of the million-dollar question. And really, from everything we study, from a functional perspective to a molecular perspective, it appears as if these cells and tissues are, in fact, acquiring a more youthful state. So just by that definition, until we have a true molecular definition of cellular age, we're working on this. This looks like a true rejuvenation. So moving cells and tissues back from an age state back to a younger state through a kind of reprogramming. So we published this in the early 2000s, looking at both muscle and liver, and then collaboratively with our colleague at Stanford, Tony Weiss-Corey, we started looking at the brain and neural stem cells. And then over the many next years, many other tissues were examined in these heterochronic parabiotic pairs and found to show restoration of youthful function in the spinal cord, the pancreas, the heart, brain, vasculature, et cetera, et cetera. So many tissues have been studied and have been shown to have this rejuvenating effect by exposure to young blood. And of course, this doesn't escape the popular imagination. This is from a episode called Blood Boy from Silicon Valley, the satirical HBO comedy, in which this older entrepreneur is getting an infusion of blood from this young, healthy male with the idea that this is going to help him live longer and healthier over time. But our question is, are there more practical interventions? We're obviously not going to be parabiotically pairing humans that would restore youthful function to old cells. And this circles back to, again, the main topic of today, which is exercise. And Hippocrates had written, eating alone will not keep a man well. He must also take exercise. Of course, Hippocrates, I guess, didn't know about caloric restriction, so he might have said eating less will not only keep a man well, but he must also exercise. But clearly, there has been this idea of the benefit of exercise to health and longevity for centuries and millennia. So this is just an article that I like from the New York Times, because I too, like Dan, I'm a runner, I run marathons, and according to the article, an hour of running may add 7 hours to your life. So I like the math, so I guess if I just keep running, I can just basically achieve what they call escape velocity and just keep running and running and running forever. I don't know if that will work, but I'm working on it. But this is a slide from an NIH program called MotorPack, which is the molecular transducers of physical activity, which asks this fundamental question that Dan raised and that I am talking about, which is, why does exercise do this? I mean, what is it that happens when we exercise that reduces our susceptibility to all these diseases, whether it's lung cancer or diabetes, heart attack, and cardiovascular disease, bone health, muscle health, brain health? I mean, all of these are well known, but really have little idea at this point on what the real molecular transducers are of physical activities. And this is actually, Dan referred to this, so this idea that muscle does not only move our body, move our joints, but it's actually a secretory organ. So muscle secretes things, and it secretes things locally, and it secretes things into the blood. these so-called myokines that Dan mentioned, that are these positive regulators of metabolism, but they have positive effects on fat, on liver, on pancreas, on bone. And so the question really is, are these myokines, are what's secreted by muscle, are these the mediators? And the more we exercise, the more positive things are secreted from our muscle, both locally and then into our blood, that might be mediating these beneficial effects. So I want to just talk about how the evidence we have that these might be acting on stem cells. And some of the first evidence of this actually came from Rusty Gage's lab at the Salk Institute, in which they found that exercise, just in the form of wheel running, had this profound effect on stem cells in the brain, so-called neural stem cells, leading to so-called enhanced neurogenesis, the formation of new neurons. And these are neurostem cells in the area of the brain that are important for learning and memory. And it was clear that exercise can stimulate these neurostem cells and this enhancement of learning and memory as perhaps one of the most profound things you can do to enhance brain stimulation and neurogenesis. So a few years ago, we asked the question, does exercise actually rejuvenate old muscle stem cells? So the way we did this experiment is we put mice on one of these running wheels and we exercised them for a few weeks. And then we, again, looked at their muscle injury responses. And this is work done by a former MD PhD student in the lab, Jamie Brett, and a current postdoc in the lab, Marina Arjona. And what we found was really quite striking. So again, mice run on a running wheel for 3 weeks, and then we injure the muscle. And we measure on the y-axis here is just an index of the regeneration. So how well the muscles regenerate in young versus old mice. What was interesting is we then had some mice not exercise and some mice exercise. Exercise had very little effect on the young mice in terms of this muscle regeneration. But in the old mice, which have an impaired regeneration compared to young, exercise almost restored this regeneration back to a young level. So in a way, similar to what we saw with the parabiotic experiments. So clearly exercise was having this impact on this repair. And we looked at actually at the muscle stem cells and we asked, what are the genes that are turned on in these old stem cells in response to exercise? And this is just a complicated plot looking at lots and lots of genes. But what you see high up here, the gene that was the most highly induced in response to exercise is this gene, cyclin D1, which is a very famous gene in terms of regulating cell proliferation and so-called cell cycle. So we ended up doing a lot of experiments, which I won't go into, in which we either reduced the levels of cyclin D1, and we showed that impaired the ability of exercise to enhance muscle repair. So clearly, this gene, its induction is important for this exercise response. And equally importantly, if we maintain cyclin D1 levels high in old muscle stem cells, we could enhance the repair of muscle in the old animals. So clearly, cyclin D1 is playing a pivotal role in this stem cell population and in response to exercise. So this is just our kind of working model of how this looks. So here you have young mice and old mice, and we look at their muscle stem cells from these hind limb muscles. Cyclin D1 levels go down with age, and with that decline, you see an impairment of this regenerative response. But with exercise, you can induce, even in the old animals, this cyclin D1 back up to a more youthful level, and you can get improved regeneration. So what we don't know is if this is the same pathway that works in other stem cells, but this is what we're interested in studying and how this is regulated. And again, if there are factors in the blood that can be identified that can lead to this response, they could potentially be turned in some kind of therapeutic, not the exercise pill, as Dan said, but at least therapeutics that could potentially enhance different aspects of stem cell function that change with age. So I'm just going to end with two more interesting pieces of data from these studies. And the first is something that will be familiar to anyone who trains and then stops training, and that is the effect of exercise is transient. We run these mice on a wheel for 3 weeks. And again, we're looking here at kind of a stem cell function. Immediately after 3 weeks of exercise, that function is improved. Then over the course of 1 week, 2 weeks, as we all know, that benefit declines. And so once you stop training, you know that your fitness goes down fairly quickly and goes back down to baseline. So that's no surprise, but the molecular pathways and the cellular pathways kind of parallel what we know has happened physiologically. And then the last piece of data I want to share is if we took old mice and we transfused them with blood, either from another old mouse that had not exercised, or from an old mouse that had exercised, we asked, what does the effect of those plasma transfers have on stem cell function? And what we found, this is just shown here on the graph on the right, is that the effects of transfer of plasma from non-exercised mice is much less compared to the exercised mice. So this just says that there actually is something in the blood of mice that exercise that can be transferred by a blood transfusion into another animal to produce the benefit of that exercise. Just confirming or supporting the idea that there are factors within the blood that are mediating this beneficial effect that potentially could be identified and could potentially be translated into some kind of therapeutic. Okay, so let me just end by again talking about this idea of aging as this is the fountain of youth, of course, and can it be reversible? Can it be reversed? Is aging reversible? We know that aging is malleable in the sense that we can slow aging, we can delay the processes that change, but from this kind of data, as well as other data from other kinds of experiments, there's increasing interest in the idea that at least aspects of the aging of cells and tissues can be reversed at least transiently. And I think from a point of view of therapeutics, one can really imagine that being translated into tissue repair. So the time of a bone fracture or the time of a skin wound, could you introduce some kind of therapeutic in this way that could restore youthful function to the older stem cells to enhance the repair, at least of that tissue during that time? So I think that's where we think of this beginning and then extending that more to longer term treatments in terms of delaying the onset of age-related diseases.